All right, so I haven't gone through all the exam questions in this exam. I would say that this one was actually the one that requires you to write a lot. Okay, so they said I explain the process of proton secretion for maintaining acid-base balance in the kidney. So proton secretion is simply you talking about the role of hydrogen ion in maintaining what acid-base balance in the kidney. So as simple as it seems, if they say the whole kidney, you must actually start talking about all the parts of a nephron, starting from the glomerular filtration to the proximal convoluted tubule to the loop of Henle to the distal tubule and collecting tubule, and also talk about buffer systems involved. You get it? So let's start it quick since it is long. We said that maintaining acid-base balance is a critical function of the kidney. All right, and secretion of the protons, that's what hydrogen ion, it actually plays a key role in this process. Okay, so here we are looking at how this kidney manages proton secretion to maintain acid-base balance. So let's start from glomerular filtration, which is here. So I'll use this picture more. Okay, so the glomerular filtration, blood is filtered through the glomeruli into the kidneys, and this will now allow things like water, electrolytes, and small molecules, including bicarbonate and hydrogen ions, to pass into the tubule. Okay, so they pass into the tubule, but know that we are focusing on hydrogen ion, okay, because we're talking about the protons. So all of this will pass into the renal tubules, while larger molecules and blood vessels will be retained back in the bloodstream. So that's why when you are having large molecules like proteins passing, we now say what? This barrier, glomerular barrier, is not functioning well. Okay, so that's it for filtration. Then for proximal convoluted tubule, this is a proximal convoluted tubule, this one. Okay, so the reabsorption of bicarbonate. Okay, so here in the proximal convoluted tubule, there's reabsorption of bicarbonate. So bicarbonate is reabsorbed back into the bloodstream. Carbonic anhydrase converts bicarbonate and hydrogen ions into what? Carbonic acid. So you are combining this to form this. Okay, so this carbonic acid then dissociates to form water and carbon dioxide. You get? So you have what? H₂O. The remain was CO₂. Do you understand? So this CO₂ can now diffuse into tubule cells where it recombines with water to form the carbonic acid, which will then dissociate to give you back the carbonate and the hydrogen ion again. Alright? So this bicarbonate is what reabsorbed into the blood, and hydrogen is secreted into the tubular lumen. Alright, everything we're talking about is this one. I'm trying to get it. Yeah, this reaction basically. Okay, this reaction basically. So let's have it. Okay, there's also ammonium secretion. So glutamine is metabolized in the proximal convoluted tubule to produce ammonia. That's what NH4 plus, and also bicarbonate. So this NH4 plus is secreted into what? The tubular lumen, and bicarbonate is reabsorbed into the blood. Okay, so what happens at the loop of Henle? This is the loop of Henle. For the loop of Henle, there's passive transport. So the loop of Henle primarily what? Handles the concentration of urine through passive reabsorption of water or solutes. Okay. Now, for distal convoluted tubule and collecting ducts, we have lots of work happening here. So what happens? There's proton secretion. So in the distal convoluted tubule and the collecting ducts, hydrogen ions are actively secreted into what? The tubular lumen by the intercalated cells. So this whole process is facilitated by what? Hydrogen ATPase. So this is a proton pump that actively transports hydrogen ions into the lumen against concentration gradients. Then we have the hydrogen potassium ATPase. This is also an exchange pump that would secrete hydrogen into the lumen while reabsorbing what? Potassium ions. Okay. So there's also bicarbonate reabsorption here. Okay. So the secreted hydrogen combines with what? Filtered bicarbonate in the lumen to form carbonic acid. Okay. So now this carbonic acid can now dissociate to form what? Carbon dioxide and oxygen and water again, okay? So the carbon dioxide diffuses back into the cells and the process repeats, resulting in a net reabsorption of what? Bicarbonate and elimination of hydrogen. So this is still what happens at the proximal convoluted tubule. I think I didn't bring the picture of what happens at the distal tubule, so you guys should not really understand. But this is what actually happens at the what? Proximal convoluted tubule. You can see how this hydrogen is shot back into the tubular lumen after this carbonic acid dissociates into it, okay? And this what? Bicarbonate is reabsorbed, you get. So that's the mechanism. Then looking at the buffer systems involved, we say we have the phosphate buffer system. The hydrogen phosphate in the tubular fluid binds to what? The secreted what? Hydrogen ion. Now it will now what? Form, if you are having another hydrogen formed here to be what? H₂PO4. So if you add the hydrogen, it will now form what? Dihydrogen phosphate, which is now secreted in urine. Then we have ammonia buffer. So ammonia in the tubular fluid binds to what? Secreted what? Hydrogen ion. So if ammonia is NH3, if you're adding another hydrogen, it will now form what? Ammonia. Okay? And this will now be excreted in urine. So what is the outcome of all this secretion of protons and all of that? The combination of bicarbonate reabsorption, proton secretion, and buffer systems, all of them allows the kidney to now be able to maintain the acid-base balance, okay, by it to neutralize acid, so it will help to remove what? These protons from the body, and to reclaim bicarbonate, so it's to ensure that bicarbonate is retained and returned to the blood stream, okay. So that's a long story, but you just have to do it. Explanation of the process of what? Proton secretion for maintaining acid-base balance in the kidneys.